So they were looking at the function of globular proteins, right? So key topics we're going to look at is reversible binding of ligands is essential. What do we mean by reversible binding? Yeah. So when things bind and unbind, what kind of forces are involved? Chemical forces. Hydrogen bonding, hydrophobic forces, weak chemical forces, not covalent bonding, things like that. Maybe some ionic bonding. They're going to be typically weak binding forces are critical because things have to be able to come off and then back on. You don't want permanent binding. Some drugs are made to permanently bind to the receptors, and those are called irreversible inhibitors or so forth. But biologically, you want things that are reversible, so you can return to a normal state. Something binds to its receptor, it's going to cause some kind of signal transduction in the cell in response to some kind of stimulus, and it'll go back to some kind of ground state again. So ligand binding is often coupled to a conformational change. What is that?
Change in shape?
Yeah, change in shape of the protein. So that's where we come up with this induced fit model of ligand binding. So I think traditionally when you think of like a receptor binding its ligand, like the traditional fit was like a lock and key model, or the traditional idea behind it was a lock and key, that the things are perfectly complementary. When the key binds to the lock, it like fits perfect. You don't get any change. It's just like perfectly fitting. But the thing is, proteins aren't static like that. They're flexible structures. So what happens is you get an induced fit when a protein receptor binds to its ligand. It's going to change its shape. And if you remember, the shape of a protein is often responsible for its function. So when you have a ligand bind to the receptor, it changes its shape, which is going to change how it's going to interact with other molecules or interact as an enzyme, right? Does that make sense? So that induced fit is important for ligand binding. It's not just, when we get to enzymes, we'll see that also. In multi-subunit proteins, remember the quaternary structure of a protein involves more than one subunit, right? My example of that was hemoglobin has 4 subunits versus myoglobin, which is only 1 subunit. But in multi-subunit proteins, conformational changes in one subunit can affect the conformational changes in the other subunits. And that's where we get something known as cooperativity. So that's like if you have a ligand that binds to one subunit, it can be negative or positive cooperativity could be involved, and it's going to change the other subunits' interactions with their ligands. Does that make sense? So the example we're going to see is with hemoglobin. Once you get one of the hemoglobin subunits bound with oxygen, it increases the affinity for all of the other 3 for oxygen. So you can really saturate that hemoglobin with oxygen in a high oxygen environment. And interactions can be regulated. And oftentimes, what's it called? Robert? I forget the word.
Positive negative feedback.
Not feedback, but the kind of receptor interactions, allosteric. Remember that?
Yes.
So allosteric behavior. So we'll see allosteric behavior of enzymes and other proteins, and they're regulated. You can have allosteric effectors, positively affecting things, or negatively affecting. And what they do is they affect their affinity for the ligands or other things. So and we'll see this illustrated by hemoglobin, antibodies, and muscle contraction. They're all examples of that we're going to see. So functions of globular protein. So they can be storage of ions and molecules. So we store iron in the body is stored by a protein named ferritin. And what does myoglobin store? Oxygen. Yeah. So we store oxygen in our muscles in the form, with myoglobin, which is basically like a monomer of hemoglobin. Hemoglobin has 4 subunits. Myoglobin looks looks almost the same as just one subunit of hemoglobin. It's just one subunit. Transport of ions and molecules. We'll see hemoglobin, serotonin transporters, defense against pathogens. We have antibodies. Antibodies. You guys know the shape of antibodies? Who's in immunology? Just two of you? Immunology? Learn all about antibodies in immunology. Cytokines are also involved in the immune system also. Muscle contraction, actin and myosin. We're going to see how that works to make our muscles get shorter when we're trying to pull on something or move. And then biological catalysts, chromatrypsin and lysosome. Anybody know what chromatrypsin is? It's a protease. So we'll learn about that specific class of proteases, I think in the next chapter, the chapter after I'm confused. I don't know. And lysozyme is an enzyme, if you ever do work with bacteria, where you purify recombinant proteins or plasmids out of bacteria, you'll oftentimes use lysozyme, and it's used to lyse bacteria. It breaks down their cell wall.
That the stuff that's in lysosomes?
Not lysosomes, but it is found in your tears. So it catalyzes the breakdown of the cell walls, and that's one of your biological defenses against infection in your eyes. Is the lysozyme found in your tears. It's also found in eggs. So reversible and transient processes of chemical equilibrium. We've all seen this before, right? You have A plus B is in an equilibrium process, and it forms the complex AB, right? You guys remember this from general chem? And we can write out rate equations for the rate that it forms a complex and the rate that it falls out of a complex, right? And then you can come up with equilibrium constants for that too, right? Do you guys remember how to do that? I'll show you this too. So you have A plus B goes to AB. That's governed by K1, right? You guys remember that? And B, or AB, will break down, that's K minus 1, and form A plus. So the rate at which you get the formation, so that's the rate of formation is going to be equal to this, K1 times the concentration of A times the concentration of B, right? You guys remember that? And then the rate of this association, I don't know if I'm writing this thing right, is that's going to be equal to K minus 1 times concentration of the complex. And what happens at equilibrium? Yeah, they're equal. So you set these two equal at equilibrium. You set the two equations equal. You have K minus 1 times A times B equals, wait, not K minus, K1 times A times B equals K minus 1 times AB. Those are equal, and we put all the chemicals on one side and all the rate equations on the other. You can do it either way. One's like an association rate constant, and the other is not rate constant, association equilibrium constant. The other is the dissociation equilibrium constant. They talk about both in here. I think you're familiar with the book that makes it kind of confusing, where they use the same symbols for multiple things. So we put all the K's on one side. AB divided by A. And which one is that? That's the
It's capital K.
K. Is that an association or dissociation? And then if you just flip it over, it's the KB, right? minus KDs are purple. Huh? Hyperbolic? Well, yeah. They're hyperbolic. Anyways, that's how you get these equilibrium constants. I may have flipped these. I'll check in a minute. We'll see. So, and we already talked about ligands, right? A molecule that binds to a protein is called a ligand. They can be other proteins. Don't just assume that they're all small molecules. When we're talking pharmacology or drugs, often it's a small molecule that's considered a ligand. But they can be other proteins. A region in the protein where the ligand binds is called the binding site, and that's pretty self-explanatory, right? The binding site is where the ligand is going to bind. The ligand binds via the same non-covalent forces that dictate protein structure. So there are weak forces, your hydrophobic forces, your hydrogen bonding, what else? You're not going to form, well, yeah, salt bridges, but I thought you meant disulfide bonds.
Oh.
Yeah, so things like that. And they allow the interactions to be transient because it doesn't take a lot of energy to break them apart. And in fact, they'll be in equilibrium, but there'll always be a certain number of molecules disassociating and a certain number that are going to associate, depending on what these constants are when they're at equilibrium. So, binding quantitative description, and I used the different numbers. They were using Ka and Kd. For some reason, I must have taught K1 for the forward reaction and K minus 1 for the reverse. Doesn't really matter what it's called, as long as you understand that the rate constants. So the kinetics of such process is described by the association rate constant, or K1, or the dissociation rate constant, which I wrote up here as K minus 1. And after some time, the process will reach equilibrium. Now you're right, we had it right. Yeah. Ka is what we did solve. I guess I did it right.
Is it still K little
Yeah, K little a is what our, and that's the association rate constant. But a lot of times we're going to see things referred to as the dissociation rate constant, and that's K little d, big K little d. And it's just the reverse of this. Okay? And in fact, we've already seen this before in acid-base chemistry, right? This K little a is they're referred to as KAs also in acid-base chemistry, but it's not quite the same because the Ka that we use in acid-base chemistry is really the Kd of that proton, right? This dissociation equilibrium constant of that proton within the acid. Does that make sense? Or am I confusing the heck out of it? Here we are using a Ka in a different context. Again, so that's the confusing thing about kinetics and pharmacology is they use this Ka, and then there's the Ka for the acid equilibrium constant, and then there's Kds, and sometimes there's a K big A, and as long as you can decipher what's going on, and hopefully you understand what they're talking about. So binding analysis in terms of the bound fraction, that's what this little theta stands for, is the bound fraction. And if you look at this, you have the protein and the ligand, or the receptor and ligand complex. Over here I wrote that as AB. One of those is going to be the ligand, one will be the receptor. So your bound ligand complex, the fraction that is bound is going to be equal to the amount of bound complex divided by the total amount of receptor, right? Total amount of protein is going to be equal to the bound plus the unbound. Does that make sense? So your fraction bound is just going to be the bound divided by the total. And if you look, you can kind of take these from the equation here. We have this Ka equals AB divided by A minus little b. So you can solve for the AB.
So when they do the theta, why isn't the denominator the complex plus 1 plus the other? Why is it just complex plus 1? Like there's no plus out of the denominator in the first equation?
What do you mean, in this equation?
Yeah.
But this is, like we're talking about, this is the total number of receptors. And beta is equal to the fraction of,
Okay. So
Receptor.
the ligand doesn't count independently because it's.
yeah, because it's not what we're looking for, we're just looking for the fraction of receptors that are bound, right?
Yeah.
So total receptors is in the denominator and bound receptors in the numerator. And then we can solve for this total receptors, right? And if we switch my AB to PL to coordinate with the PowerPoint, I guess I should have did this, right? This is P and L. We can solve for this PL, right? We'll get rid of this for now. We have Ka equals that. If we solve for PL, we multiply it. Ka, okay, big A or little a? Can be confusing. Ka equal times the free protein times the free ligand equals your concentration of bound protein, your bound receptor complex. And then all they did here is substitute this in for PL, so you get this, and then you can reduce that to this. You guys need to see that, or how I got from this to this? Wish I could mark on the thing. That would probably ruin the screen. You'll see the marks there forever. So we can extract free protein out of this everywhere, right? This will just be one. You have ligand. And then we have to extract, if we extract 1 over Ka from the 1, do you see that? We can get rid of the Ka here, so it's just ligand divided, free ligand divided by the ligand plus 1 over the Ka. And what did 1 over the Ka equal? Kd. See, simple answers. Yeah, it's right there. I always ask the obvious when it's given to you in PowerPoint. And that is the equilibrium dissociation constant. So that's basically how you're going to calculate the fraction bound if you know the ligand concentration. Free ligand. So in graphical analysis, the fraction of bound sites depends on the free ligand concentration and the dissociation rate constant. And you can solve to see what fraction bound you're going to need to have, to calculate the KD. The KD is always the fraction at which you have 50% of the receptors bound. So experimentally, the ligand concentration is going to be known, and the KD can be determined graphically or via the least squares regression, or 50% at which you're bound. You notice this is hyperbolic, and it kind of is saturated. You'll never get 100% bound, but you approach 100%. And the example of this is the oxygen binding of myoglobin. So it's like this rectangular parabola, and you get saturation quite quickly as you increase the concentration or partial pressure of oxygen. So when a ligand is a gas, binding is expressed typically as partial pressure of the gas. So the interaction strength can be expressed as the association binding constant, or per molar, or the dissociation constant, which is molar, which is 1 over the Kb, or 1 over the Ka. And the interactions binding free energy, or the standard state free energy, is in units of kilojoules per mole. And the definitions, we've heard this, right? The free energy change, or Gibbs free energy concentration change. Oh, I can't talk. Gibbs free energy equation, right? Change in free energy is going to be the change in enthalpy subtracted by the temperature in Kelvin times the change in entropy. So negative free energy changes are favorable for forward reaction, right? Positive, typically the reverse reaction is going to happen. So the relationships, free energy, so if you're changing concentrations, these are this little circle here, that means standard state, right? Anybody know what standard state is? What conditions those are? It's like 1 atmosphere, 1 molar concentration of everything. I think the temperature is what, 220 degrees C, or not 220 degrees C, like 293, or
Isn't it like 300?
I thought it was like 20 or maybe 25. Yes, it's like 298.
Is it like 20 Celsius?
25 degrees Celsius, right?
So then it's,
Or somewhere around
and
there.
then it would be 270
Exactly.
or 280.
Something like that. But very physiologically relevant conditions, right? Everything in our bodies at 1 molar. Right? No. That includes concentration of hydrogen ions, right? 1 molar. What would be the pH of 1 molar hydrogen ion concentration? You don't know? Negative log of 1. What is that? Oh, 0, right. So that's not conducive for life. So you always have to change this when oh, see, I have 25 degrees C is the standard state conditions. But the relationship, you can convert from standard state free energy, because you'd be given charts with the free energy, standard state, for different reactions, and that's going to be equal to negative RT natural log of the Ka, or the get rid of the negative, right? You just have natural log of the Kb, because that is the inverse of the Kb. So that means strong binding interactions is when your Kd is really, really small, right? Does that make sense? Smaller the Kd, the stronger the binding. So you're going to see antibodies have really strong binding characteristics, and their Kds are in the molar, or in the picomolar to nanomolar concentrations. Weak binding is where you have much higher Kds, up to anything greater than 10 micromolar would be considered weak binding. And these are some protein dissociation constants. We have avidin is one of the strongest. So that is, anybody know what avidin is? Did I talk about that before? It's used as a reagent in labs everywhere. Purified out of egg whites. It's the reason if you drink raw eggs on a regular basis, you can end up with a vitamin deficiency. Because it's got such strong binding to biotin, which is one of your B vitamins. So it'll bind up all your B vitamins and cause you to have a biotin deficiency if you eat raw eggs constantly. But we don't do that. We typically cook our eggs, which, what does cooking eggs do? Denatures the protein, so it doesn't allow that so much. So yeah, this is one of the, and avidin has 4 binding sites for biotin, and it's the storage molecule for biotin. Insulin receptor, if you look at that binding interaction, it's 1 times 10 to the minus 15. So what is that? So 10 to the minus 9 is micromolar, right? And then 12 is nano, so the picomolar Kd concentration. Insulin receptors, 10 to the minus 10. Anti-HIV immunoglobulins are again 10 to the minus 10, but 4 times 10 to the minus 10. Nickel binding protein, which is going to be your polyhis tags, bind nickel really strongly. That's 10 to the minus 6, so it's not that strong. And then calmodulin is a protein that binds free calcium. That's a 10 to the minus 12. Lower of it. So, specificity. We already talked about this a little. Proteins typically, they do have for the most part, highly specific binding interactions with certain ligands. There's always going to be non-specific binding interactions, but they're going to have really big KDs or really low affinity, right? And that's why we use things like albumin to block plates when we're doing ELISA. We're blocking any non-specific protein binding interactions with the plate, because the antibodies we use in ELISAs and things like that are going to bind to these non-specific protein binding sites, but we want to use other proteins that aren't going to interact with the assay to block those sites. That's why we use milk and Western blots or albumin and ELISAs to block those non-specific binding sites. We typically use a high concentration of those proteins to block those sites. So the high specificity can be explained by the complementary of the binding site to the ligand. And this is, Emil Fisher came up with this lock and key where things just fit perfectly together. So they're going to bind and then they fit perfectly together, and then I guess the only change in function would be the function where this binding site is, where they're bound together. There's no change in shape of these proteins. But the induced fit is what we find things are more likely, or not more likely, you can actually look at the structure of proteins that are bound to, their ligands, and you see structural shifts. Conformational changes occur upon the ligand binding. That's called the induced fit. Both the ligand and the protein can change their conformations. So like I said, the ligands don't have to be small molecules, they can be other proteins, and they can both change their shape. Here you get an interaction, and they both change to this active or inactive state. And we'll see that in enzymes, and there will be active enzymes when they're bound to their effector, or inactive enzymes when they're bound to their effector. It all has to do with this conformational shift that we get from the binding interactions. So now we're going to shift to the globulins, such as myoglobin and hemoglobins, globulins, tomato. So protein side chains lack affinity for oxygen. It's going to be hard to get oxygen to bind to just side chains of proteins. So that's why we need to have some sort of, what's it called? Got something attached to a protein. Some sort of, what's it called? I'm bad at words today. It's Monday. I hate Mondays. So you have an apo enzyme and a holo enzyme. We talked about this already? No. A cofactor, coenzyme. But they give it a different name when it's covalently attached. Yeah, what's it called? Never mind. Talk about it. But so what we do is we capture the oxygen molecule with a heme that is protein bound. You guys know what heme iron is? It's like the best way to absorb iron. Normally, if you eat a lot of spinach, you don't really absorb that much iron. You know, it's super high in iron. But the best way to get iron absorbed in through your gut is when it's already complexed with this porphyrin ring known as heme. It's just a structure that's like a cyclic structure, and it coordinates the heme. So myoglobin is the same main oxygen storage protein. Hemoglobin is the circulating oxygen binding protein. And this is the porphyrin ring and heme. So you have these nitrogens around iron. If you guys remember, iron is one of those transition metals, so it's got all those places for electrons to coordinate into those empty shells. And you have the electrons here on the nitrogen, which will coordinate this iron and keep it in the heme ring. It's like this planar ring, the iron binds in its center. So not only do you get coordination from the heme ring that's involved, in this case in myoglobin, you also get a nitrogen from one of the imidazole rings and a histidine that coordinates with the iron. And then the other binding site on that iron can be occupied by oxygen or carbon monoxide or something like this. So the fact that this imidazole is involved with this iron might be the site where you get that induced conformational change upon binding, right? This is going to bind in the iron at the same time the oxygen will. It's a place where you get that interaction. And carbon monoxide has a similar size and shape as oxygen and can fit to the same binding site. So it can bind also. Carbon monoxide binds over 20,000 times better than oxygen because the carbon in carbon monoxide is filled with a lone electron pair that can be donated to the vacant d orbitals in the iron. Protein pocket decreases the affinity for carbon monoxide, but it still binds about 250 times better than oxygen. So that's why if you've got a garage attached to your house and leave your car running, you can get carbon monoxide poisoning, or if you got a leaky gas heater. We don't have this problem too much in Florida.
So the 20,000 times, that's not like.
So that's just the heme alone, not in
Not in the
protein, not in the globulin or globin. I would say globulin, globin. So that's just the free heme. So carbon monoxide is highly toxic, right? Because it competes with oxygen, so it's going to be a competitive binder to oxygen. In fact, it has a much higher affinity for the heme and myoglobin or hemoglobin. So the function of myoglobin, hemoglobin, mitochondrial cytochromes, which contain this heme group also, they're involved in oxidative phosphorylation. So that's, if you have carbon monoxide in the mix, it's not going to allow that oxygen to be that electron acceptor, right? Because it's not going to chemically react the same way that oxygen will. In fact, they use carbon monoxide as a reducing agent, don't they? You guys know what industrial process they do that? They teach you that in general chem. So to purify iron, you take iron ore and you have to give it electrons. They use carbon monoxide in those big stacks. They make by cooking coke with a small amount of oxygen, generates carbon monoxide. That carbon monoxide then is a reducing agent that will reduce the iron ore down to free iron, or metallic iron. So it doesn't work well in our heme as an electron acceptor. It's actually an electron donor in the industrial process. But I digress. But it binds so well is due to this structure. Oxygen is going to, remember, the electrons are off to the side, whereas in carbon monoxide, they're directly 180 degrees across, so it fits really well into that heme structure, has a super high affinity. So heme binding to the protein affects carbon monoxide versus oxygen. And this is in the protein itself. There's not quite as much room as if it was free to get that type of structure. So oxygen is better, but still carbon monoxide still has a higher affinity. So could myoglobin transport oxygen? I guess it could, right? You could have circulating myoglobin rather than hemoglobin. The problem with that, I mean, what it does, so we have is this change in oxygen pressure, partial pressure of oxygen. So in the lungs, we have a high partial pressure of oxygen, right? So we'd be able to probably saturate that myoglobin in the lungs with oxygen. It'd be easy to saturate. But as we move to the periphery, that partial pressure of oxygen drops a little bit. So you'd have to get it way down here to dump off the majority of that oxygen. Otherwise, if you go from like 5 to, 2.5, you really haven't released that oxygen in the periphery. So myoglobin itself is not designed well to be a good transporter of oxygen because it can bind it really well, but you really have to lower that partial pressure of oxygen for it to really release that oxygen. It's really good at binding the oxygen, but not good at releasing it unless the pressure is really low. So you could lower the affinity of myoglobin, and that would help if this curve shifts to the right. It flattens this out a bit, right? But you still need these drastic shifts. But for effective transport, you have to vary, the affinity has to vary with the concentration of oxygen. Like I was telling you, it's a hemoglobin itself has 4 subunits. And remember I was telling you, once you bind to one oxygen, that increases the affinity for the other oxygens, right? So it exists normally at this, if it doesn't have any oxygen around, or doesn't have any oxygen bound, it's going to be in this low affinity state. So it's like the hemoglobin itself has 2 types of affinities. There's a low affinity state and a high affinity state. With a high partial pressure of oxygen, you get binding, and that increases the affinity of all the other subunits, so it's going to pick up even more. And that's called positive cooperativity. That's what we see in hemoglobin. That's why it's able to pick up so much oxygen in the lungs, it shifts to the high affinity state. Whereas when you get to the periphery, you start to lose that oxygen. Once you start losing the oxygen, it shifts it to a low affinity state where you'll be able to dump off even more oxygen in the periphery. So oxygen is a positive allosteric effector of hemoglobin. There's some negative allosteric effectors of hemoglobin also. So just thinking about things that happens in the periphery during metabolism, can you think of any things that might negatively affect oxygen? Right? Because you want to shift it to a low affinity state in the periphery. So what's generated in the periphery that might shift hemoglobin to a negative state or low affinity state? Carbon dioxide. Yeah. So that's one of the negative allosteric effectors in hemoglobin. It lowers the affinity of hemoglobin to oxygen when there's a higher concentration of carbon dioxide. What else gets generated during metabolism? Carbon dioxide adds to this too. When you dissolve carbon dioxide in water, what happens? Yeah, carbonic acid, and then that'll disassociate one of those protons. So acidic conditions also shift hemoglobin to that low affinity state. So you get this combination. There's another negative regulator, but it's not associated with metabolism per se. But the pH, or the high concentration of, or if you lower the pH, you get a high concentration of protons, and that's what happens in the periphery where metabolism is taken. Same thing with carbon dioxide. Increase the carbon dioxide concentration, increases the low affinity state. So you're able to dump off more oxygen from that hemoglobin than you would be if you didn't have these shifts in affinities. Does that kind of make sense? I think I got more pictures, better slides. So how can affinity to oxygen change? Boy, I jumped way ahead of this last slide. So binding sites, you have multiple binding sites. Hemoglobin has how many binding sites? Yeah, 4. So binding sites must be able to interact with each other, and I already told you that they do. Right? When you get binding at one site, it changes the shape of all the other sites. So they all are cooperative in their binding to oxygen. Positive cooperativity is how hemoglobin reacts to binding oxygen. It's positively cooperative. The first binding of it increases the affinity at the remaining site, and it's recognized by a sigmoidal binding curve. What we mean by sigmoidal, right, it's like this S. So at low oxygen concentrations, it kind of follows this low affinity state, but then as you increase the partial pressure of oxygen, boom, you shifted to this high affinity state, and you totally increase the affinity of oxygen. So negative cooperativity would be the opposite. It's when you have the first binding event will reduce the affinity at the remaining sites. And this is just an example, I think your book shows you, of not hemoglobin that has 4 subunits, but something that has 2 subunits. We have the binding site here, and this red part is more unstable. It's not, that's where you get a conformational change, and it'll flip-flop back and forth to the high binding site and to the low binding site, depending where that equilibrium in the cell shape, or not the cell shape, the protein conformation lies. Once you get binding at one site, though, if it stabilizes this, right, it's less stable, but it's still stable, and it's going to increase the affinity for the other site. Now it'll be positive quantitative. Whereas you could also have a case if it binds the ligand at one site, you could make it more stable and block that site too, right? So that would be negative quantitative. They don't talk about this in this. So cooperativity. Cooperative proteins have multiple ligand binding sites, so the association equilibrium constant becomes this, where you have the number of ends, and that's going to be the number of binding sites, right? And theta is going to be changed to this, where you have a free ligand to the nth power plus the kb on the bottom. So taking the log of both sides gives you the Hill equation. You guys ever seen the Hill equation? That's the equation that describes that sigmoidal shape of things. Where you have the log of theta divided by 1 minus theta equals n, the log of the ligand concentration subtracted by the log of kd, and n is known as the Hill coefficient, and that is the degree of cooperativity you get between those subunits. N minus 1, I can't see that, n equals 1, you have no cooperativity, right? N equals 1, this is just going to be your fraction of bound substrates. So the Hill plot of cooperativity. So here we see. So and it combines the low affinity state to the high affinity state. If you have myoglobin here, you have a Hill. The Hill constant here is 1, so you have no shift in affinity based on the concentration of oxygen. Where partial pressure of oxygen does change, you get this Hill coefficient here, and that's an NH of 1 versus 3. So there's two models of cooperativity, concerted versus sequential. And right, so concerted means that, so once you get one binding event, concerted means that they're all going to shift to the other affinity state, right? Increase. Or it could be sequential, where we get binding of 1, that increases the affinity of 1, and then you bind 2, and that'll increase the affinity of the next one, and so forth. Whereas, so that would be sequential, right? Concerted is when you bind 1, they all become high affinity, and then they switch. So it's either or. Two different models. Cooperativity is a special case of allosteric regulation. I love allosterism. So allosteric proteins, you have binding of a ligand. The one side affects the binding properties of a different site on the same protein. Can be positive or negative. Homotrophic is the normal ligand of the protein is an allosteric regulator. And we see that with hemoglobin as a homotrophic allosteric regulator. Oxygen is a homotrophic allosteric regulator of hemoglobin. And then you can have heterotrophic, which is a different ligand affects the binding of the normal ligand. And we already talked about 2 different ligands to hemoglobin that are heterotrophic negative allosteric regulators. They were carbon dioxide and hydrogen ions, right? Cooperativity is going to be equal positive homotrophic regulation. And that's what we see with oxygen. It's positive homotrophic regulation is positive cooperativity for hemoglobin. So hemoglobin is a tetramer, meaning it has 4 subunits of 2 subunits. So there's alpha 2 and beta 2 is typically what you see, and then there's all kinds of mutant subunits too that you might have in certain mutations, like Sickle-cell is one of the mutant subunits. But each subunit has a similar shape to myoglobin. This is myoglobin, here's your beta subunit of hemoglobin, and they have very similar structures. There's a heme group, and they have this like almost heart-shaped like structure. And they also have similar in sequence. This would be like some bioinformatics where we're comparing the sequence of myoglobin versus hemoglobin alpha, hemoglobin beta, and all the same amino acids are highlighted, where they're all 3 share the same Same when we don't have diacetyl. We do get subunit interactions in hemoglobin. And these cases here, these alpha helices interact together, here, and then here is interacting with that subunit, here's interacting with this subunit. So you get these contact points where the individual subunits are going to interact with each other. And there's some detail with the sum. So you have a histidine interacting with aspartic acid, and here you have a lysine interacting with an aspartic. They don't have this one labeled, but it's got to be aspartic acid or something forming a salt bridge, right? And that's on a beta subunit and an alpha subunit. So lysine, you guys remember what charge lysine is? Lysine accepts protons. It's got the side chain has got a pKa of around 10.5 or so. So at physiologic pH, the lysine is going to have a positive charge, which is going to be able to form a salt bridge with an aspartate, which generally at physiological pH has that negative charge, right? So that's where you get a salt bridge interaction. And these show you that some of those interactions. Arginine can have a positive charge and interacts with an aspartate. This is alpha 2 interacting with the alpha 1, arginine interacting with an aspartic acid again. And then you get the beta 1, carboxylic acid, the C terminus of that beta 1 interacts with the lysine. And then you have aspartic acid interacting with the histidine on itself, and so forth. So it wasn't aspartic acid, that was the C terminus that I was pointing at, right? So the C terminus itself is interacting with this lysine. So you'll hear this when we're talking about allosteric regulation or allosteric proteins. They exist as two different states: a tense state and a low affinity and a relaxed state. So the tense state, you have more interactions, so it's more stable. So it's tense, right? More interactions, more stable, and has a lower affinity for oxygen. We're talking about the tense state of hemoglobin. So lower affinity for oxygen, more interactions between the subunits. Once you get one oxygen binding, it's going to shift that to the relaxed state. Fewer interactions between the hemoglobin subunits. And it has a higher affinity for oxygen. It's more flexible, so it's relaxed, right? Oxygen binding will trigger that tense state to convert to an R state, conformational change. That conformational change from the T to the R state involves breaking the ion pairs between the alpha 1 and beta 2 interface. Let's look at that ion pair, alpha 1 and beta 2. That looks like the lysine right in the C terminus. So we're getting that salt bridge. And this, wait, am I going the total wrong way? I never know what these buttons do. So this is a picture of the structure of hemoglobin in the tense state versus the R state. You can see in the tense state, there's the alpha 1 and beta 2. Here's the somewhere along here. You can see the C terminus and that histidine interacting. Whereas you see it's different here in the relaxed state. You don't see this alpha 1 and beta 2. Instead, you see these histidines not bound up in the center. And conformational change is triggered by oxygen bonding. So if you remember, there's this histidine on this F helix, as it's known as, that coordinates the histidine from that F8, it's the 8th subunit on that F helix, coordinates with the heme iron. Once you bind oxygen, if you notice, I don't know if you can tell from this picture, it's kind of puckered in towards the histidine. When the oxygen binds, it kind of goes from a puckered shape, this heme, to a more planar shape. And And that causes a shift in the position of this helix, which causes that overall conformational change. This is shows you like how big that conformational change can result in all the subunits. So pH effect on oxygen binding to hemoglobin. So actively metabolizing tissue, we already talked about this, generates hydrogen ions, lowering the pH of the blood near the tissues relative to the lungs. So the hemoglobin affinity for oxygen depends on the pH, right? Hydrogen ions to the hemoglobin. Hydrogen ions bind to the hemoglobin and they stabilize that T state. Protonates histidine 146, which then forms a salt bridge with aspartic acid 94 and leads to the release of oxygen in the tissue. So just the fact that you have metabolism going on due to the fact that we're alive, right? You generate higher concentration of hydrogen ions. It protonates this histidine, which changes that overall shape, leading to that release. It's a way to get more oxygen dumped off where it needs to be, where the metabolism is occurring. pH difference between the lungs and the metabolic tissues increases the efficiency of that transport. This is known as the Bohr effect, that pH effect. And this shows you where you get that added proton. So here you have a histidine. With the proton added, gives this a positive charge, so it's able to form this salt bridge between beta-1 histidine and beta-1 aspartic acid, stabilizing that in the tense state, making it more stable. You have a salt bridge there, tensing up that hemoglobin, thus lowering the affinity of oxygen. So as you increase the oxygen concentration, at different pHs, you get different affinities. And it shifts the curve to the right, as the pH lowers, meaning the hydrogen ion concentration is increasing as the pH lowers, right? Is that right? Everybody confuses that pH negative log, so as hydrogen ion goes up, pH goes down. And we see here, this curve shifts to the right. The best way to understand that is, as it shifts to the right, it just means you need more oxygen to get the same level of binding. If you look at it, it changes that the KD goes from, if we were expressing it in kilopascals, it's like 3 kilopascals to maybe 3.75 to about 4 kilopascals. And as you increase that KD, it lowers the affinity. So hemoglobin and carbon dioxide export also. So carbon dioxide is produced in metabolism. And in tissues and muscles, it must be exported. 15 to 20% of the carbon dioxide is exported in the form of a carbamate ion on the amino terminus residue of the polypeptide structures of hemoglobin. So this is the case where you actually get a chemical interaction with the amino terminus, reacts with the carbon dioxide itself, and you form a carbamate. The form of carbamate yields another proton, which adds to the Bohr effect, and the carbamate forms an additional salt bridge, which will stabilize that hemoglobin in a tense state. So the rest of the carbon dioxide is exported as dissolved bicarb, formed by carbonic anhydrase, which also produces another proton, adding to that Bohr effect again. And this shows you the reaction at the amino terminus. So you have the amino terminus of hemoglobin, reacts with the carbon dioxide, and forms this carbamate ion, giving up a proton that was originally on that amino group. And then this will form a salt bridge. And one of the other negative allosteric regulators, negative heterotrophic regulator of hemoglobin function, is this chemical compound here, 2,3-bisphosphoglycerate. So this is a negative allosteric regulator. And what it does is it fits down in the center portion of the hemoglobin molecule and makes hemoglobin have a lower affinity for oxygen. This comes into play. This is why there's a big difference between fetal hemoglobin and maternal hemoglobin, or normal adult hemoglobin. Fetal hemoglobin has a lower affinity for this 2,3-bisphosphoglycerate. So it doesn't bind to this negative allosteric effector as well. So fetal hemoglobin will have a higher affinity for oxygen because it doesn't have as much 2,3-bisphosphoglycerate bound to it. So at the placenta, the mother's hemoglobin may have a lot of 2,3-bisphosphoglycerate, but when it comes close to the fetal hemoglobin at the placenta, you can get transfer of oxygen from that mother's hemoglobin to the fetal hemoglobin because the fetal hemoglobin doesn't bind this as well, so it has a higher affinity for oxygen. So, thus, the fetal hemoglobin doesn't have the T state stabilized as much. This shows you where that 2,3-bisphosphoglycerate binds to at that center pocket, stabilized by these imidazole groups on the histidine. Right? And this shows you it again down in the central cavity, stabilizing the 10th state of hemoglobin. And this shows you how it's going to shift how it shifts the binding curve. So this is bisphosphoglycerate at 5 millimolar at sea level. This is bisphosphoglycerate at 8 millimolar at higher altitudes. So it shifts 8 millimolar to the right. So you're able to dump off more oxygen at a higher altitude also. So spectroscopic detection of oxygen binding to myoglobin. So the heme unit's a strong chromophore, right? That's why our red meat's red and our blood is red too. That absorbs a lot of light. Ferrous form, Fe2 without oxygen, has an intense sorbent band of 429. The oxygen binding alters the electronic properties of the heme and shifts that position of the sorbent band to 414. So the binding of oxygen can be monitored via UV vis spectrophotometry. The oxyhemoglobin in the venous blood appears purplish, and oxyhemoglobin is red. That's why you always see it drawn like that, right? And then sickle-cell anemia is caused by one mutation. One glutamic acid, or glutamate, number 6, is mutated in the code and it's substituted for the valine on the beta chain of hemoglobin. Can anybody tell me the difference in structure between the valine side chain and the glutamate side chain? Fine to look it up. Which is negatively charged, which would likely be found on the exterior of a globular protein, right? Interacting with the solvent of water, right? So what about valine?
Valine has no charges.
It's an aliphatic hydrocarbon, right? So it'd be more likely to be found in the interior, right? So the sickling of blood cells occurs typically in low oxygen. What happens is that valine is out there in the aqueous phase and it doesn't want to be. So it causes a tendency for that hemoglobin. You get interaction between the molecules and those valines will want to interact with each other. So they reduce the amount of surface area that the water is exposed to it. So it'll polymerize and you get these crystals that form. And they misshapen the normal shape of a red blood cell, and that's where sickle cell disease comes from, because it kind of elongates them, because these long crystals that crystallize in those blood cells, and it makes a sickle-like shape. It looks like one of the sickle that Grim Reaper grabs us with, or you cut the wheat or grass with, right? It has that sickle shape, and you can't deliver oxygen too well. It doesn't fit through the capillaries. What evolutionary advantage do you think you get from that sickle cell mutation? Huh? Dying?
That's not an advantage.
No, it's not an advantage. And normally you only get the sickle cell disease if you don't have any normal beta hemoglobin. Remember, in your chromosomes, you have 2 copies of each chromosome, So a lot of times you'll end up with, maybe you'd be a carrier of sickle cell disease, but you'll never get it because you have one bad beta hemoglobin, and another one would be normal. In that case, that sickle cell mutation is way diluted. But it has an advantage. And that is when the,
Pumps carrying less oxygen.
well, it's not that it carries less oxygen. So if you're infected by a parasite that can live in your blood, decrease the amount of oxygen, that's when you get the sickling. It affects how well that parasite will survive in your blood. So sickle cell, being a sickle cell carrier gives you resistance to malaria. So that's why it's a bad trait to have that sickle cell anemia. It makes it probably less likely to be able to carry that gene on it, constantly getting sick anytime you run or anything like that. But that's carried on through evolution because it has that good set trait that the people who are carriers are able to survive malaria more often than people that don't have that sickle cell mutation at all. So, now we're on the immune system, and I never took immunology. It's a tough class from what I hear. Is it? So, the two types of immune system: cellular immune system, which targets own cells that have been infected. Also clears up virus particles and infecting bacteria. Key players are macrophages, killer T cells, and inflammatory T cells. And you have your humoral immune system, which targets extracellular pathogens. Can also recognize foreign bodies, and it makes these soluble antibodies. We've seen antibodies before. We use them in the lab all the time. They have that one shape. Right? What shape do they have? Yeah, they're a Y shape. Y shape. And it's made from 4 smaller proteins. And there's they're cross-linked together, something like that. The the antigen binding sites are at these tips of the Y. And then this is your FC region. This would be your FAB region at the 2 antigen binding sites. And these are you have long chains and your short chains also. And these FAB regions at the ends where you have the antigen binding sites are known as your variable regions, because those are the regions. And the genes that code for antibodies that can shift around a lot. You make multiple different types of antigen binding sites, and eventually you'll make one that'll bind to that antigen, and that gets produced by, or they don't say the B lymphocytes, right? And then the memory cells will store the antibody coding region and start producing them when it's in the presence of the pathogen. So antibodies bind the fragments displayed on the surface of invading cells. Phagocytes, specialized cells that eat the invaders. Macrophages are large phagocytes that ingest the bacteria that are tagged by antibodies. So here in this case we have a virus coated with antibodies, or IgGs. There's different classes of antibodies. IgGs find in the blood. There's IgMs, IgAs. IgYs are what you're going to take proteins class and purify that from the A. We don't find IgYs in us, per se. So the macrophage is coated with Fc receptors. And if I told you this is the Fc region, this is always constant. And like a human antibody, you're always going to have the same, they all look the same. So they're always going to be able to bind to the Fc receptor. When you get a coated pathogen bound to the Fc receptor or multiple Fc receptors, it causes the macrophage to engulf that virus or bacteria, pathogen, and it'll digest it, and then it'll display it even further after that. So vertebrates also fight infection with the soluble antibodies that specifically bind the antigens. And I didn't explain what antigens were, right? So antigens are are basically the ligands for these FAB fragments or antigen binding regions of the antibody. So they're the ligands that the antibodies recognize. And they're the substances that stimulate the production of antibodies, typically macromolecular in nature. You can make antibodies to small molecules. Those are, you have haptens, or what those are known as. Typically, you do that by adding that hapten to a larger molecule and to produce those antibodies. It's how you can produce antibodies to T8 or the THC breakdown products, so you can have antibodies that recognize, drug metabolites. That's how your drug tests work. So typically, macromolecular in nature, recognized as foreign by the immune system, and they coat proteins of bacteria and viruses are a lot of times antigenic. Surface carbohydrates of cells or viruses. That's why if you give somebody the wrong blood type, it's the carbohydrates on those cells are not recognized as self, so your immune system will attack them also. Also, antibodies are proteins that are produced by B cells and specifically bind to the antigens. Binding will mark the antigen for destruction or interfere with its function. So in the lab, you can make antibodies that will actually interfere with the protein's function to try to correct disease. If you have a mutant protein that's causing a problem, if you give antibodies or just the FAB fragment, you can actually block the function of that bad protein. And then if you only deliver the FAB fragments, you won't have the FC there, so you won't generate an immune response to that bad protein, although you might want to. It's on your strategy to treat that disease. Binding will mark the antibody for destruction, and a given antibody will bind to a small region, so that's the epitope of that antigen. So you might have heard monoclonal versus polyclonal antibodies. You guys ever heard those? What's the difference?
Monoclonal or identical, and then Sickle-cell.
Yeah, monoclonal means they come from one clone or one cell, and they all recognize the same epitope of the antigen. Whereas polyclonal antibodies, where they're from many blood cells, so you have many blood cells making antibodies that recognize multiple epitopes on that antigen. And you can purchase monoclonal antibodies. They make them by they inject a mouse or a rabbit. They did monoclonal rabbit antibodies too. With the antigen that you want to make antibodies to, then they take the spleen out, fuse those to cancer cells, and then they grow them on a 96-well plate after they've diluted the fused cells down to where you'd only get one cell per well or so. And then they test the supernatant for activity of producing antibodies to the whatever antigen you want. And if you have a hit, you grow those cells up and it'll produce that antibody for as long as those cells will stay alive, which can be a long time if you freeze them periodically. So one antigen can have multiple epitopes. So you can have a virus coat that is a protein, and there could be multiple sites on that protein that can generate an epitope that different antibodies will recognize. So polyclonals, where you just immunize an organism, generates an immune response against that antigen, and there's many different cells producing antibodies to multiple epitopes, and that's why it's polyclonal. Many different cells, and you can coat that antigen with antibodies. So IgG is composed of 2 heavy chains and 2 light chains, and it's also composed of a constant domain and a variable domain. Constant domains are like here, actually here, and variable domains are at the end or the FAB fragment. So variable domains of each chain make up the antigen binding site up here, and the domains contain regions that are hypervariable, specifically the antigen binding site that confers high antigen specificity. And this is probably better than my drawings. I put too many disulfide bonds there. There's only one between the light chains and the heavy chains. This is your FC region and your FAB region. antigen binding site, which contains the variable domains of the heavy and light chains. And this is an actual 3D structure of an IgG bound to a carbohydrate. And kind of hard to tell.
Why would it come down there?
Because this is one of the FAB binding regions.
What's the red compared to the blue?
The F's, yeah. I don't know. Doesn't look right, does it? I would think the red would be the F, this would be where your light chain, and the blue would be the heavy chain, right? So what is the carbohydrate? Yeah, I would think the carbohydrate would bind here, if I'm interpreting the picture right. But you would expect them to put it like that, right?
Yeah, I don't know.
So they don't really say in the figure legend. Yeah, 2 shades of blue are the heavy chains, and the light chains are shades of red. So these should be your FAB fragments and your FC region. I don't know why they have a bound carbohydrate. So that antibody doesn't, the carbohydrate isn't the antigen, it's binding. Antigen binding causes significant structural changes to the antibody. So this is your induced fit again. It's a conformational change with no antigen bound, but with the antigen bound but not shown, and this is the antigen on that antigen binding site. You can see the blue regions have shifted significantly in relation to this red region, which they keep it in the same spot pretty much. So that's the induced fit. Antibodies have an induced fit also. So antibody specificity is an important analytical reagent. And this is an example of an ELISA, enzyme-linked immunosorbent assay of some sort. Huh?
Yeah, there's something to say, I think that's what it is.
Enzyme-linked immunosorbent assay. This actually might be a Western blot too, it's hard to tell. But some solid support where you're looking at proteins. So this is a plate, it'd be an ELISA, or it could be a membrane for a Western blot. You have an antibody, primary antibody you're using specific to the analyte you want to detect and measure its concentration. Then you're going to have a secondary antibody, and it recognizes the FC fragment of the primary antibody, and it's often conjugated to some sort of enzyme, most likely horseradish peroxidase or alkaline phosphatase, which will then react with a substrate to produce either some kind of color, fluorescence, or light as a reaction product. And in this case, it's an ELISA, and you get this yellow color with increasing concentrations of that protein that you want to detect. And so you quantify it. Taking proteins class, we have you measure concentrations of the IgY you extract from the A. So picogram quantities that you get down to. So protein interactions modulated by chemical energy. The use of chemical energy, such as ATP, can cause conformational changes in proteins generally required for their function. So some conformations, proteins will flip back and forth between different conformations, but if you add energy to it, it may take energy to get to one conformation over another, right? So especially in motor proteins, you can use ATP to control the movement of cells and organelles within the cells, and allows for spatial and temporal regulation of the interactions. And we'll see with muscles, it requires ATP for the actin and myosin to get those changes in shape. So muscle fiber is a large, single, elongated, multinuclear cell. Each fiber contains about 1,000 myofibrils, which are like this. And these fibrils can be broken down into individual sarcomeres, where you can have an A band and an I band, an M line and a Z disc, where you get these overlapping structures that kind of overlap each other and get overlap more as the muscle contracts and overlap less as it relaxes, right? myofibrils contain thick filaments of myosin. And these are your myosin. So it's a dimer, and then these dimers will coil on each itself, and you have these large myosin molecules with all these head groups, and the heads contain actin binding sites. Not just actin binding sites, there's also sites, just the actin binding sites, ATP binding sites also, or ADP binding sites. The myofibrils contain thin filaments of actin. Remember, I think we talked about, when we were talking about structural protein, somebody mentioned actin as a structural protein. It's fibril, that's why it's F-actin, is fibril, but it's made up of all the, it's a fiber of all the G-actin monomers. So G-actin subunits or globular actin binds together and forms these fibrils. And that myosin head will interact with the actin filament. Myosin binding sites on the actin binding sites on that myosin head. And it's this interaction, that doesn't work. Ignore this, these used to just contract and stuff. For some reason my animation is not working. So the contraction, so this is a relaxed one, and as you contract, these myosin head groups kind of walk their way down these actin filaments and cause this band to get shorter, and that overall changes the, shortens that muscle fiber. It's going to squeeze it together, or pull it together. So and there's a cycle, an actomyosin cycle that causes this muscle contraction. And it occurs through a series of conformational changes to the protein structure, and they're due to binding, hydrolysis, and release of ATP and ADP. So the cycle's 4 steps. First, the ATP binds to myosin, and this, when ATP binds to the myosin, it causes that myosin to disassociate from the actin. So when myosin is bound with ATP, that causes that myosin head to have a lower affinity for the actin, and it causes it to disassociate. Once you hydrolyze the ATP, hydrolyzing the ATP converts the ATP into phosphate and ADP, and that causes another conformational change of the myosin. And then that myosin reconnects to the actin filament at a different location. You get the release of the phosphate. Release of the phosphate is known as the power stroke, and that's where the myosin returns to its initial shape, shifting the actin filament relative to the myosin tail, and then it releases the ADP. And it's, then available to bind another ATP and release from the actin. Burns that ATP to ADP and phosphate, binds again, and when you release the phosphate, it shifts the head again, releases the ADP, another ATP comes in. This one was working earlier when I checked it. Oh, it didn't work here. It worked earlier. Maybe I'm not doing something. Anyways, this one was a nice little animation I found online. I'll find a link to it. But it shows you each step. So, now we all know muscle just doesn't constantly contract, right? So how is that regulated, you guys know? That means A and P, I don't know if they cover basic core bio.
They do it in physiology.
Physiology?
Yeah.
Anybody know what controls the contraction of muscle?
Monoxide.
Well, the neuron fire, so you get an action potential from a motor neuron. It causes release of what from the sarcoplasmic reticulum, releases calcium into the cytosol muscle. That calcium then binds a tropomyosin, or troponin. There's a calcium binding site on the troponin, which is bound to tropomyosin. Without calcium there, this troponin blocks the actin binding sites of on the actin, right? Blocks the myosin binding. I get confused. So on the actin blocks the myosin binding sites. And when it binds calcium, it kind of lifts, and when you bind calcium to the troponin, it lifts this troponin away from that actin binder, the myosin binding site on the actin, allowing the head to then complete its cycle where it binds ATP, pops off, ATP gets cleaved to ADP, falls back down, binds the next step. The phosphate disassociates, you get a conformational change where it kind of pulls itself down this actin filament. Then once the ADP disassociates, it stays there, and then an ATP associates, causing it to release from the actin filament, which is kind of spelled out here, right? I already covered the regulation. So the regulation of the availability of myosin binding sites in the actin is regulated by troponin and tropomyosin. And so that avoids that continuous contraction of muscle. So nerve impulse triggers the release of calcium from the sarcoplasmic reticulum, causes a conformational change to tropomyosin-troponin complex, exposing the myosin binding complexes on the actin, allowing myosin to work its way down so long as ATP is available for it to go through those conformational changes. That's it for chapter 5.